Yeah. Welcome back, everyone. And today we discuss sex determination in humans. Before we start, let's remind ourselves of how gender is controlled, of how a male becomes male and how a female becomes female. Right? We know that if we're talking about a male will have the gonosomes which are XY. Gonosomes, which is a terminology we use to describe sex chromosomes, right? So we know that the gonosomes of a male, they are XY. But when we talk about a female, we know that the gonosomes of a female are XX, right? So if this was in a karyotype, we know that we find these gonosomes in position number 23 in our karyotype, that is in the final position, right? So if we have a larger chromosome and a smaller chromosome, that will tell us that it is XY. The larger chromosome is the X chromosome and the smaller chromosome is the Y chromosome. Therefore, that person will be male. And the reason we'll give in that scenario is that this person has got one X chromosome and one Y chromosome. But when we talk about females have got two X chromosomes. So in our karyotype, in position 23, which is the final position, we will see two large chromosomes. So the larger chromosomes are XX. And if we were asked to identify the sex of that individual, we would say that individual is female, and the reason we'll give is that the person has got two X gonosomes, which are our sex chromosomes, right? So we know that in order for a person to be male, the chromosomes have to be XY. In order for a person to be female, the chromosomes have to be XX, right? So now, let's discuss these gonosomes again. If this female, for example, has got the XX gonosomes, it tells us that one of these gonosomes came from the paternal origin and the remaining gonosome came from the maternal origin. That is how it always works, right? So one chromosome is received from the paternal origin and the other chromosome is received from the paternal origin, right? So if we're saying that one of these chromosomes was received from the paternal origin, it was received from the paternal origin through the sperm cell of the father, because we know that in the sperm cell we've got a part that we call the nucleus, and that nucleus is going to be haploid, simply telling us that it has got half the number of chromosomes. And in its nucleus, it will have a gonosome, just one gonosome, because it is now haploid. So in a karyotype, there's going to be one chromosome per position. So in position 23, this sperm cell will either have an X chromosome or it will have a Y chromosome, right? And then in the maternal origin, these chromosomes are going to come from the mother's ovum, right? The same applies to the ovum. The ovum is also going to be haploid. This tells us that in each position in the karyotype of this ovum, there's only going to be one chromosome each. So in position 23, the story remains the same. There's only going to be one chromosome, and it will always be an X chromosome. So the gonosome that is going to be produced by the female parent will always be the X gonosome. But when we talk about the sperm cell, the gonosome produced can either be X or it can be Y, right? So now let's try to reason what we're trying to explain here. So if this sperm cell, let's say it is carrying an X gonosome this time, right? Fertilizes this ovum. So in position 23 of this sperm cell, there's going to be an X chromosome. In the ovum, in position 23 of this ovum, there's also going to be another X chromosome. So if fertilization occurs in position 23, there's going to be this X chromosome from the paternal origin, and there's going to be another X chromosome from the maternal origin, therefore resulting in a baby that is female. It therefore tells us that if the sperm cell of the father contains an X chromosome, then the baby that is going to be born is going to be female, simply because the mother will always contribute an ovum that has got an X chromosome in position 23. But it may happen that the sperm cell that is produced carries a Y chromosome. If it carries a Y chromosome, it tells us that the sperm cell now in position 23, it will contribute a Y chromosome. But remember, the ovum only carries an X chromosome, so the ovum will also bring an X chromosome there. So if the sperm cell is carrying a Y chromosome, then the resulting baby is going to be a male, simply because it received this X chromosome from the mother and this Y chromosome from the father, which is the paternal origin. So generally, this is how sex works, right? And we need to be able to explain this phenomena in words, and we call this phenomena sex determination. This is how the sex of the baby is decided. It is mainly decided by the sperm cell, which can contribute either an X chromosome or an X chromosome, right? Let's put that into words. Point number 1, female have got XX chromosomes. This is an obvious statement. We already know this, that the chromosomes of females are XX, and we know that in our karyotype we find these chromosomes in position 23, right? And therefore, the ovum that is going to be produced will only have an X chromosome. This will always be the fact. An ovum can only carry an X chromosome. This is simply because the female only has XX chromosomes. So the ovum that is going to be produced will only carry one of those X chromosomes, right? But when we talk about males, we know that the chromosomes of the males are XY. Again, this is another statement that we know, that the chromosomes in male are XY, right? Now let's talk about the sperm cell that is produced. The sperm cell that is produced by males can have an X chromosome, or it can have a Y chromosome. So it can be one of two. It will be an X or a Y chromosome. This is in a sperm cell. But with an ovum, it will only be an X chromosome, because the chromosomes of the female are XX. But with the sperm cell, the chromosomes can be X or they can be Y, and that is simply because the chromosomes of a male are XY, right? Now, let's talk about fertilization. We know that fertilization simply describes the process where the sperm cell fuses with an ovum. After this sperm cell has fused with an ovum, it is going to form a single diploid cell, and this diploid cell is what we call a zygote, right? Now, let's talk about this sperm cell. We know that the chromosome that is in this ovum will always be an X chromosome. Always. The chromosome will be an X chromosome, right? But let's say this sperm cell now is carrying an X chromosome. So when fertilization occurs here, the chromosomes in this zygote are going to be XX. And if the chromosomes in this zygote are XX, it tells us that this zygote is going to develop into a baby that is a female. And this was determined by the sperm cell that was carrying the X chromosome, right? But it may happen that the sperm cell is carrying a Y chromosome. If this sperm cell is carrying a Y chromosome, then when fertilization occurs, the zygote that is going to be formed now is going to have the chromosomes XY. And this zygote will develop into a male, right? Simply because we know that the chromosomes of a male once more are XY and the chromosomes of a female are XX, right? And that is why we try to explain with this statement. So if a sperm cell carrying an X chromosome fertilizes an ovum, then the child will be female, and that is because she will have XX chromosome, one from the paternal origin and one from the maternal origin, right? But if the sperm cell is carrying a Y chromosome and fertilizes the ovum, this is what is going to happen. Then the chromosomes of the child are going to be XY. And we know that XY chromosomes are chromosomes that are found in males. That is why we say that the baby is going to be male and the chromosomes are going to be XY. That is because we know that the chromosomes of males are XY, right? So with this in mind, we can therefore say the following statement. The father determines the sex of the child. It is the father that determines the sex of the child. That is simply because the father is the one that produces a sperm cell with either an X chromosome or a Y chromosome. If the sperm cell of the father has got an X chromosome and fertilization occurs with the ovum, then the baby is going to be female because she's going to have XX chromosome. But if the father produces a sperm cell with a Y chromosome, then the baby produced there is going to be male. And that is simply because the child is going to have XY chromosome. Obviously, in the case where the sperm cell produces the Y chromosome, then the X chromosome can only come from the maternal origin. The Y chromosome is only present in the sperm cell, so it comes from the paternal origin, right? But there is always a 50% chance that a child born will either be male or a female, right? Let me try to make an example. Let's say we've got a female. We've got a family, and in this family, the husband and the wife have given birth to 3 kids, right? Let's say all the kids that they've given birth to are females. They have 3 female children, right? And let's say the female now is pregnant. The mother is pregnant with the fourth child, right? And the question asks, what is the chance that the child that is going to be born is either going to be male or is going to be female? The chance that the baby is going to be male or female will always be 50%. It doesn't matter how many male children or how many female children have been born in that family. The chance that the baby is going to be male or female will always be 50%. And we're going to show that by means of a genetic cross, right? So let's try to look at this example number 1. Example number 1 says, use a genetic cross to show how gender in human offspring is determined by the sex chromosome of the parents. So we're supposed to use a genetic cross here to show how the sex is determined by the sex chromosomes of the parents. Obviously, if we say parents, we're talking about a male that is crossed with a female. These are our parents, right? And we know that the chromosomes in males are XY. Therefore, this is our male genotype. And we know that the chromosomes in females are XX, and therefore this is our female genotype, right? Let's try to show this by means of a genetic cross. And we know that when we do our genetic cross, top left, we simply write P1, and those are parents of the first generation. In front of P1, now comes phenotype. Now comes phenotype, and the physical appearance here is simply a male that is crossed with a female. This is our phenotype, right? What about the genotype, which is the genetic makeup of an organism? The genetic makeup of a male will be XY, and the genetic makeup of a female will be XX, right? Below genotype now we write meiosis. In front of meiosis we write nothing. Below meiosis now comes gametes. Now comes gametes. Right? With gametes, we simply separate the genotype by comma. So it is going to be X, Y, that is crossed with X, X. Below gametes now comes fertilization. Now comes fertilization. And let's use our Punnett square. So it is going to be 3 boxes sideways and 3 boxes downwards. Right? So this is our Punnett square. Top left, we write gametes. So in front of the word gametes, we're going to write the gametes of the male parent. So it is going to be X first and then Y, written in separate boxes. And then below the word gametes, we're going to write the first X in the female and the second X in the female. So in this box, our genotype is going to be the X from the father and the X from the mother. So it is going to be XX. In this box right here, it is going to be the Y from the father and the X from the mother. So it is going to be XY. And then in this box right here, it is going to be X from the father, X from the mother. So it will be XX. And in this box right here, it is going to be the Y from the father and the X from the mother. So it will be XY. Right? Below the word fertilization, we're going to write F1. And we know that F1 simply stands for offspring of the first generation. So in front of F1, we're going to write genotype. We're going to write genotype. And this is the genotype of the offspring that we produce through this cross. So it is going to be X, 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 Y, X, Y. Right? And then below genotype, we come to write phenotype. And this is the physical appearance of the offspring. The first offspring, XX, this is female. XX, that is female. This one, XY, that is male. This one, that is male. So it therefore tells me that my phenotype is going to be 2 females. There's going to be 2 females and 2 males. There's going to be 2 females and 2 males. So if we try to look at the percentage chance here that the offspring born is going to be female, it is going to be 2/4. It is going to be 2/4 multiplied by 100, so the answer will be 50. If it is males, we've got 2 males out of 4 offspring total over 50. So it is going to be 2/4 multiplied by 100, so it will also give us 50%. So this tells us that the chance that the next baby that is going to be born from a couple is always 50. Right? So the chance that the next baby is born is going to be female, it is 50%. The chance that it will be male, it is 50%. And the sex of this baby is controlled by the male parent. Right? Let's continue. Now let's come to discuss sex-linked genetic disorders. Right? These are disorders that are linked to sex. Specifically here, we're talking about the gonosomes. We're talking about the sex chromosomes, right? Here we're going to discuss alleles that cause disorders. But these alleles that cause these disorders are linked to the sex of an individual. So these alleles are carried on the X chromosome. So the Y chromosome does not carry any alleles that cause genetic disorders, right? So these alleles that causes genetic disorders are only carried on this X chromosome, right? So in simple terms, the alleles causing a sex-linked genetic disorders are mainly carried on the X chromosome. So they will not be carried in the Y chromosome, right? Now, with these disorders that are caused by alleles that are carried on the X chromosomes, there are 2 that we need to know by heart. And this is what we call hemophilia and color blindness. We've got these 2 disorders. Disorder number 1, we call it hemophilia, and disorder number 2, we call it color blindness. Now, what is hemophilia? Hemophilia is a genetic disorder that causes the blood to not be able to clot. When the blood is not able to clot, it simply tells us that this person is going to bleed severely without stopping because the blood cannot clot, right? And this is hemophilia. It is a genetic disorder. And we also have another genetic disorder that we call color blindness. It is also called daltonism. Color blindness simply refers to the condition where an individual is unable to tell the difference between colors. So these are genetic disorders, right? Now let's discuss them one by one. Let's talk about hemophilia. Now, hemophilia is caused by a recessive allele small letter h. And even if we did not include the word recessive allele here, if we only said hemophilia is caused by the allele small letter h, we would have known that the allele that is causing hemophilia is recessive, because we know that once we represent alleles as small letters, we're simply saying that this allele is recessive, right? But remember, we said that hemophilia is a sex-linked genetic disorder. Therefore, this allele, which is small letter h, that causes hemophilia, has to be carried on the X chromosome, because we are saying that the X chromosome is the one that carries the allele that cause genetic disorders, right? So, therefore, the allele that is going to cause hemophilia, we are going to write it as X to the exponent of small letter h, simply showing us that the allele causing hemophilia is carried on the X chromosome, and this is how we denote it. So it is going to be the X chromosome to an exponent of small letter h, which is our recessive allele that causes hemophilia, right? So if this is our recessive allele, therefore the allele that is normal can only be X to the exponent of capital letter H. So the allele that is normal is going to be capital letter H. But also, since this is an allele that is related to hemophilia, and hemophilia is a sex-linked genetic disorder, it is also carried on the X chromosome. Hence, we write it as X to the exponent of capital letter H, right? Now let's talk about daltonism, which is color blindness. Color blindness is caused by a recessive allele small letter d. Now the story is the same. If only they told us that color blindness is caused by an allele small letter d, we would have known that the allele causing color blindness is recessive. Because once we write an allele as a small letter, we know that this allele is recessive, right? Now, since color blindness is a sex-linked genetic disorder, it tells me that the allele that causes color blindness is also carried on the X chromosome. So how is this D that causes color blindness carried on the X chromosome? It is carried as X to the exponent of small letter d. We know that this is the allele that is going to cause color blindness, because we said that color blindness is caused by the allele small letter d. And therefore, the allele that is normal will therefore be X, representing the X chromosome, to the exponent of capital letter D. That will be our normal allele. Now, it is very important for us to know that hemophilia and color blindness are sex-linked genetic disorders, and therefore anytime we show genotypes or we are required to do genetic crosses involving hemophilia and color blindness, it is important to remember that they are sex-linked genetic disorder, and therefore their genotypes must be expressed with the sex chromosomes at the same time, right? So we must know color blindness and hemophilia by heart. Any other sex-linked genetic disorders that are not color blindness and hemophilia, they will tell us that such and such is a sex-linked genetic disorder. But with hemophilia and color blindness, it is not actually necessary for them to tell us that they are sex-linked genetic disorders. We must know it by heart. Right? So let's continue. Now I've tried to show the different genotypes for hemophilia and the different genotypes for color blindness, right? Starting with the males and ending with females, right? Now let's remember that we said that the allele that causes hemophilia is an allele small letter h. But since hemophilia is a sex-linked genetic disorder, this recessive allele small letter h must be carried on an X chromosome, because we said that the alleles causing sex-linked genetic disorders are only carried on the X chromosome. Therefore, it is indicated as X to the exponent of small letter h, the X representing the X chromosome, right? But since we know that the gonosomes of a male is XY, that is why genotype number 1 here, with genotype number 1, this one, we said that a male that is normal, so this male does not have hemophilia, his genotype will be X to the exponent of capital letter H and Y. We included X and Y simply because these are the male gonosomes, right? And capital letter H here is normal, so this person is going to be normal, right? You will note that no allele is represented on the Y chromosome because the Y chromosome does not carry any alleles in this case, right? Let's go to number 2. So this is the normal male, but a male that has got hemophilia, his genotype is going to be X small letter h capital letter Y, right? So this person, this male, is going to have hemophilia. Hence we say this is a hemophilic male, simply saying that this is the genotype of an individual with hemophilia. Simply because we know that hemophilia is caused by a recessive allele small letter h, and this male has got the recessive allele small letter h without any capital letter H, simply because the Y chromosome does not carry any alleles. So if this male only has one recessive allele, this male is going to suffer from hemophilia automatically, right? So with males, it is only needed that you have only one recessive allele in order for you to suffer from hemophilia, right? Let's go to female. We may have a female that is homozygous dominant, and we know that the word homo simply tells us that the alleles that we're going to use are going to be the same. Hence, this female has got X, capital letter H, X, capital letter H. And since capital letter H is an allele for being normal, this female, we're going to describe her as being normal, right? But let's go to the second female. The second female has got X, capital letter H, X, small letter h, and we say that this female is normal. How is she normal? Since hemophilia is caused by a recessive allele, and this person is heterozygous, hence she has got the capital letter H, which is normal. It tells me that the effects of this small letter H, which causes hemophilia, are going to be masked, and therefore the person is going to be normal. Remember, in simple terms, when we combine a capital letter and a small letter, the capital letter always wins. And this is a type of dominance that we call complete dominance. So the allele for being normal here completely dominates the allele for hemophilia. So this person is going to be normal. But this is a female that we refer to as a carrier. What do we mean when we say this female is a carrier? We're simply saying that this female has got an allele that causes hemophilia, but its effects in this female are masked. But she is capable of having an offspring that has got hemophilia simply because she carries this recessive allele with her. So it is possible for her to pass on this allele for hemophilia to one of her offspring, and the offspring can actually suffer from hemophilia as a consequence, right? But the female herself, she does not suffer from hemophilia because she is heterozygous, and the allele for normal here completely dominates the allele that causes hemophilia. So she will only be a carrier, which is normal, right? But for a female to suffer from hemophilia, she must have two recessive alleles, right? So when this female has got two recessive alleles, that is X small letter H, X small letter H, this is the only condition where this female will now suffer from hemophilia. Hence we call her a hemophilic female. This is simply because we know that for the recessive trait to be expressed, there must be two recessive alleles. And since females have got XX gonosomes, she can only suffer from hemophilia if each of her X gonosomes carries the recessive allele for hemophilia. If she only carries one, she becomes a carrier. For her to be sick, she must carry all two. This brings a stark contrast to our male here. If we compare this male and this female, in order for males to suffer from this genetic disorder, which is hemophilia, they only need one recessive allele. But in order for females to suffer from hemophilia, they will need two recessive alleles. And this is simply because females have got two X gonosomes, but males only have one. So if a male inherits one recessive allele for hemophilia, he will suffer from hemophilia. But a female needs two, because if a female only has one recessive allele, she will be what we refer to as being a carrier. Now, this very same scenario applies also to color blindness, since color blindness is also caused by a recessive allele. So whatever I said here also applies to color blindness, right? Let's continue. Now, let's look at example number 2. Example number 2 says that hemophilia is a sex-linked disorder that causes excessive bleeding due to a lack of blood clotting protein. Like I said, when you have got hemophilia, your blood does not clot, so you bleed continuously, right? And it is actually dangerous since you will lose a lot of blood. May probably result in death if not treated, right? So the recessive allele responsible for this disorder is represented by X small letter H. Again, the reason why we represent this allele this way is simply because hemophilia is a sex-linked genetic disorder. And when we write alleles to represent sex-linked genetic disorders, we must always include the sex chromosomes, right? But again, as I've already said, with hemophilia, it is an absolute necessity for us to know that hemophilia is a sex-linked genetic disorder without being told. Consequently, we must also know the allele responsible for causing hemophilia and the normal allele thereof, right? So let's try to answer these questions. 2.4.1. 2.4.1 says that show by means of a genetic cross the possible genotypes and phenotypes of the parents and children. If a man that is suffering from hemophilia and a woman who is a carrier of this disorder decide to have children. Now, the man that is here, they're saying that this male suffers from hemophilia. But the female, they said that the female is a carrier. When we say that a female is carrier, it tells us that this female is normal. So the female is normal, just that she has the allele for hemophilia, but she is normal, right? So that is what we mean when we say she's a carrier. But let's try to look at the possible genotypes for this hemophilic male. We know that for a male to suffer from hemophilia, he only needs one recessive allele. So the only possible genotype for the male will be X small letter h, Y, right? But for this female to be a carrier, she is normal, but she has the recessive allele that causes hemophilia. So therefore, the only possible genotype for this female can only be X capital letter H, X small letter h. That is the only possible genotype for this female because we said that she is a carrier, right? Let's do our genetic cross. So top left, we know that we write P1. In front of P1, we know that it is going to be phenotype. This is our physical appearance. And we know that they said it is a man with hemophilia. So we are going to write hemophilic male. Hemophilic male. We can also write male with hemophilia. It is the same thing, right? And we're going to cross a normal female. We know that they said that she's a carrier, but being a carrier means that she is normal, right? Below phenotype now we write genotype. We write genotype, right? So the male with hemophilia, we know that the only possible genotype for a male with hemophilia is X small letter h, Y, crossed with this normal female who is a carrier. So it is going to be X capital letter H, X small letter h, right? Below genotype, we know we write meiosis. We know we write meiosis. And in front of meiosis, we do not write anything. So below meiosis now comes gametes. Now comes gametes. And we simply separate the gametes by comma. So it is going to be X small letter h, comma, Y, crossed with X capital letter H, comma, X small letter h, right? Below gametes now comes fertilization. Now comes fertilization. And we show fertilization by means of a Punnett square, so which is 3 boxes sideways, 3 boxes downwards. So we're going to draw our Punnett square like so. Here we simply write gametes, and in front of the word gametes we write the gametes of the first parent, so it is going to be X small letter h and Y. And below the word gametes we write the gametes of the second parent, so it is going to be X capital letter H, X small letter h. So in this box it is going to be X capital letter H, X small letter h. In this box it is going to be X small letter H, X small letter h. In this box it is going to be X capital letter H, Y, and in this box it is going to be X small letter h, Y, right? After fertilization now comes F1. This is our offspring of the first generation, and in front of F1 we write genotype. We write genotype. We simply copy our genotypes there. So it is going to be X capital letter H, X small letter h, X small letter h, X small letter h, X capital letter H, Y, X small letter h, Y. Below the word genotype now comes phenotype. Now comes phenotype, right? Now, the first female here with this XX chromosome, she's going to be normal. But because she is heterozygous, she is a carrier. But being a carrier means that she is normal, right? The second female here has hemophilia because she has got 2 recessive alleles, right? And then the third child, which is the first male, is normal because he has the normal allele here. And the fourth child, which is our second male, suffers from hemophilia because he has the recessive allele. So we're going to describe each and every one of them because they are all different, right? So we're going to say one normal female. One normal female, right? And then we've got one hemophilic female. One hemophilic female. And then coming to this one now, one normal male, one normal male, and then finally one hemophilic male, one hemophilic male. And that is our genetic cross Done, right? But the question had two questions. So let's look at the second question. It says that explain why this disorder affects mostly males in a family. So it is males who are going to suffer from hemophilia more than females. This is a factual statement. And the way we answer this, number one, we are going to note that hemophilia is carried on the X chromosome. That has to be our first statement. That hemophilia is carried. The allele for hemophilia, which is small letter H, is carried on the X chromosome. So the allele now becomes X small letter H, right? And then we also have to note that males only have one X chromosome and one Y chromosomes, right? So because males only have one X chromosome and one Y chromosome, it tells me that a male only needs one recessive allele for hemophilia in order to suffer hemophilia, right? So again, the way we answer this, we're going to say that the allele causing hemophilia is carried on the X chromosome. That has to be our first statement. Number two, we have to note that a male has got one X chromosome and one Y chromosome, right? Point number three, we're going to say that a male only needs one recessive allele, which is X small letter H, in order to suffer from hemophilia, right? But females, on the other hand, females have got XX chromosome, so they've got two X chromosomes. So a female, in order for her to suffer hemophilia, she needs two recessive alleles for hemophilia, but a male only needs one. And because a male only needs one, it tells me that it is easier for a male to suffer from hemophilia than females. Because if a female only has one recessive allele, then she becomes a carrier. But if a male only has one recessive allele, then automatically he suffers hemophilia. So that is how you are going to word your answer, right? Let's continue and do one more example. It says that the inheritance of fur color in cats is sex-linked, okay? The tortoise shell color in cats is a combination of black and orange. Let's note this statement. So this combination of black and orange, it is called a tortoise shell color, okay? The allele for black fur is represented by X, capital letter B, okay? And the allele for orange fur is represented by X to the exponent of capital letter O. So this is black and this is orange, all right? They said, hint, the sex chromosomes or gonosomes in cats are inherited in the same way as in humans. Okay, that's nice to know. 5.1. They say that a female cat with a tortoise shell is crossed with an orange male. Show the genetic cross between the two cats and determine the phenotype ratio of the F1 generation. Use X, B, X, O, and Y. Okay, so it is a female with tortoise shell. Let's remember that tortoise shell, they said it is a combination of black and orange. Therefore, the only possible genotype for this female has to be X, capital letter B, X, capital letter O. But this male that they're saying is orange, his only genotype has to be X, capital letter O, Y. These are the genotypes we're working with. So it is a tortoise shell female and a male that is orange. So let's try to perform that genetic cross. So again, top left, we're going to write P1. In front of P1 now comes phenotype. Phenotype. And it is a tortoise shell female. Tortoise shell female. Female that is crossed with a orange. That is crossed with an orange male, right? Below phenotype now comes genotype. Now comes genotype, and we've already said that the genotype for this tortoise shell female, since it is a combination of black and orange, can only be X, capital letter B, X, capital letter O, crossed with X, capital letter O, Y. Below genotype now comes meiosis. Now comes meiosis, and we already know that in front of meiosis we do not write anything. So below meiosis now comes gametes. Now comes gametes. With gametes we simply separate the genotype by comma, so it is X, capital letter B, comma, X, capital letter O, crossed with X, capital letter O, comma, Y. Below gametes now comes fertilization. Now comes fertilization. And let's show that by our Punnett square. Punnett square, 3 boxes sideways, 3 boxes downwards. Here we're going to write gametes. So let's put the gametes for the first parent here. So it will be X capital letter B, X capital letter O. And the gametes for the second parent, X capital letter O and Y. In this box right now, it is going to be X capital letter B, X capital letter O. In this one, it is X, O, X, O. In this one, it is X, B, Y. In this one, it is X, O, Y. Below fertilization now comes F1. In front of F1, we simply write genotype. This is the genotype of all the offspring that we produce. So this is our genotypes. So we're going to write X capital letter B, X capital letter O, X, O, X, O, X capital letter B, Y, X, O, Y. That's our genotype. Now below genotype now comes phenotype. Let's look at this one. This is a tortoiseshell female because it is a combination of black and orange. So this is tortoiseshell. But this female right here, she is orange. This one, this is a black male. And this one, this is an orange male. So let's write the phenotype. So it is going to be 1 tortoiseshell. 1 tortoiseshell female. 1 tortoiseshell female. And then 1 orange. 1 orange female, right? And then 1 black male, and then finally 1 orange male, 1 orange male. But the question asked us to show the phenotypic ratio. So I'm simply going to write that by PR, but you have to write the whole word. So phenotypic ratio, I'll simply include it as PR. So it is going to be 1 tortoiseshell, so it is 1:1 orange female, 1:1 black, 1 and 1 orange. So it is going to be another 1, right? Now let's try to answer the other question because there were 2. So let's try to answer this one. Right? It says that explain why male kittens can never have a tortoise shell color. Right? So we're going to start by saying again, we know that the allele for shell color, the allele for fur color is carried on the X chromosome. We need to say that statement, right? So the allele for fur color is carried on the X chromosome. But we know that in order for a cat to have the tortoise shell color, it has to be a combination of black and orange, which is carried on the X chromosomes, right? So because a male only has 1 X chromosome, it is impossible for this male to have a tortoise shell color, simply because the tortoise shell color is only formed when you have got 2 X chromosomes that are in a heterozygous condition. Therefore, 1 X chromosome will have the capital letter B and the other chromosome will have the capital letter O. But since males only have 1 X chromosome, it is impossible for them to have a tortoise shell color because it will only be formed in a heterozygous condition. And the alleles that control fur color are carried on the X chromosome. So it is impossible for males to have tortoise shell, right? Let's continue. This is our last slide. We're going to explain why males suffer from hemophilia more than females. This is generally any sex-linked genetic disorder that is caused by a recessive allele. So the way that we structured the response here applies also to daltonism, which is color blindness, or any other sex-linked genetic disorder that is caused by a recessive allele. It will apply, right? So number 1, we're going to say hemophilia is caused by the recessive allele H, which is carried on the X chromosome. So you need to tell us that this genetic disorder is caused by this allele, and this allele is carried on the X chromosome, right? Because these are sex-linked genetic disorders, right? And then you have to specify that a male only has 1 X chromosomes, whilst female have got how many? 2 X chromosomes, right? So if this is a genetic disorder that is being caused by a recessive allele, again, males only need 1 recessive allele in order to suffer from that genetic disorder. But with females, they will need 2 recessive alleles in order to suffer from the disorder. Because we know that with females, if they only inherit 1 and they have another allele which is normal, which is represented by a capital letter, then this female is going to be a carrier. And we know that a carrier is what? Is normal, right? So males will only need 1 recessive allele in order to suffer from the genetic disorder. But with females, they require 2 simply because they've got 2 X chromosomes, right? So until next time, if you like what we've had to say, consider liking and subscribing to the channel. Bye-bye.